Hello everyone, good morning one and all. Let us talk one more topic under biochemistry, that is digestion, absorption and metabolism of carbohydrates. So the objectives of the presentation. So we need to know regarding digestion and absorption of carbohydrates. Digestion, how it is happening in the mouth, stomach and intestine, and with the help of pancreatic enzymes, and digestion due to intestinal enzymes. Then under absorption, we have to mainly focusing on active transportation and facilitative transport. Under metabolism of carbohydrates, we have to know major metabolic pathways of carbohydrates. And digestion of carbohydrates, the principal sites of carbohydrates digestion or so which are the areas the carbohydrates is going to get digested? Mouth and small intestine. The dietary carbohydrates which consist of polysaccharides like starch, glycogen and cellulose, and disaccharides sucrose, maltose and lactose. So whereas monosaccharides mainly glucose, fructose and galactose. So here we can see dietary carbohydrates. Mainly we will be having polysaccharides, disaccharides and monosaccharides. And which are the areas it is getting digested? Mouth, stomach, small intestine with the help of intestinal enzymes. And finally it is getting absorbed in the bloodstream. And then monosaccharides need no digestion prior to absorption, whereas disaccharides and polysaccharides must be hydrolyzed to simple sugars before their absorption. So what we need to understand here, monosaccharides no need to having any further digestion. So like disaccharides and polysaccharides, it is getting immediately absorbed into the bloodstream. So what is the meaning of hydrolysis? It's a process of breaking down a chemical compound that involves splitting a bond and adding the element of water to the resulting molecular fragments. How the digestion process is occurring in the mouth. Digestion of carbohydrates begins in the mouth. Salivary gland secretes α-Amylase. So what is a enzyme is secreted by the salivary gland that is known as α-Amylase, which initiate the hydrolysis of starch. During mastication, salivary amylase act briefly on dietary starch in random manner, breaking some alpha 1,2,4 bonds, and starch get hydrolyzed into dextrins. How the starch is getting hydrolyzed into the as a dextrin. Then digestion in the stomach. Carbohydrate digestion temporarily happened in the stomach because the high acidity initiate the salivary alpha amylase. Digestion in the intestine. Further digestion of carbohydrates occurs in the small intestine by pancreatic enzymes. So digestion process is happening in the intestine with the help of the pancreatic enzymes. The two phases of intestinal digestion. So what is the two phases? The first one is digestion due to pancreatic alpha amylase, and second phase is digestion due to intestinal enzymes, that is like sucrose, maltase, lactase and isomaltase. Digestion due to pancreatic alpha amylase. So pancreatic alpha amylase hydrolyzes dextrin further to the mixture of maltose, isomaltose and alpha limit dextrin. The alpha limit dextrins are smaller oligosaccharides containing 2 to 5 glucose units. Then digestion due to intestinal enzymes. Enzymes responsible for the final phase of carbohydrate digestion are located in the brush border membrane of intestine. So there is a brush border membrane in the intestine. So these enzymes responsible for the final phase of carbohydrate digestion and are located in the brush border membrane of small intestine. The enzymes and the reaction they catalyze as they follows. So what is a catalyzing process? It is an action or process. The end product of carbohydrate digestions are glucose, fructose and galactose, which are readily absorbed through the intestinal mucosal cell into the bloodstream. So what is the end products of carbohydrate digestion? The end products of carbohydrate digestion are glucose, fructose and galactose. So these are readily available to absorb in the intestinal cell into the bloodstream. And here you can see this maltase. This maltose will be converting with the help of the maltase. Isomaltase with the help of the isomaltase. So sucrose is the sucrase. Then lactose with the help of lactase. And alpha limit dextrin with the help of dextrinase. Then let us talk about regarding absorption of carbohydrates. The end products of carbohydrate digestion are glucose, fructose and galactose. This you have to remember. What are the end products of carbohydrate digestion? The end products of carbohydrate digestion are glucose, fructose and galactose, which are absorbed from the intestinal lumen by two mechanisms. How it is getting absorbed from the intestinal lumen? The two mechanisms are, one is active transportation, the another one is facilitative transportation. So what do you mean by this active transportation? Active means against a concentration gradient. It is passing through against a concentration gradient from a low glucose concentration to a higher concentration. Whereas coming to the facilitative transportation with the concentration gradient, that is from higher concentration to a lower one. So the first one is active transportation against concentration gradient. It is passing from the against concentration gradient. For example, from a low glucose concentration to a higher concentration. The another one is facilitative transport with a concentration gradient from higher concentration to a lower one. So this is a intestinal epithelium, and here we can see intestinal lumen, and here we can see brush border. So in this brush border, these are getting the absorbed, and here we can see facilitative transport and active transport. Then transport of glucose, fructose. So what is a end product of carbohydrate digestion? That is like glucose, fructose and galactose and monos. And you can see some of the key points I have given here, that is SGLT, sodium dependent glucose transporter. So these are entering into the cells with the help of the sodium dependent. That is why we considering as a SGLT is indicating sodium dependent glucose transporter, whereas GLUT5 it is indicating glucose transporter. And ATP you all knows that is adenosine triphosphate, and adenosine ADP also adenosine diphosphate. Then active transportation, the transport of glucose and galactose across the brush border membrane of mucosal cells occur by an active transport. The transport of glucose and galactose across the brush border membrane of mucosal cells occurs by an active transportation. Active transport is an energy requiring process. It is energy requiring process that requires a specific transport protein and the presence of sodium ions. So this process is occurring with the help of the sodium ions. A sodium dependent glucose transporter binds to both glucose and sodium at separate sites and transport them to the plasma membrane of the intestinal cell wall. How it is getting transport? With the help of plasma membrane of the intestinal cell. In active transport, glucose is transported against its concentration gradient. For example, fluids having principles like diffusion and osmosis. Here it is with the help of the active transport. So how it is happening? Glucose is transported against its concentration gradient. The free energy required for this active transport is obtained from the hydrolysis of ATP. The free energy required for this active transport is obtained. So how the energy is getting? The energy getting from the hydrolysis of ATP, which is linked to a sodium pump that expels sodium from the cells in exchange of potassium. How it is getting? It's linked to a sodium pump that it removes sodium from the cells in exchange of potassium. One thing you remember, when there is inside of the cell either sodium or potassium should be there. If sodium is inside, potassium will be outside, or else potassium is inside, sodium will be outside. Then coming to the facilitative transport, fructose and monos are transported across the brush border by a sodium independent facilitative diffusion process which requiring specific glucose transporter, that is known as GLUT5. Movement of sugar in facilitative diffusion is strictly from a higher concentration to a lower one until it reaches equilibrium. So until it reaches equilibrium, what is happening? So the movement of a sugar in a facilitative diffusion is strictly from a higher concentration to a lower concentration. Any shifting is occurring, it is the glucose molecule will shifting from the higher concentration to the lower one until it reaches equilibrium. Then coming to the metabolism of carbohydrates, after being absorbed from the intestinal tract, the monosaccharides are carried by a portal circulation directly to the liver. So what is happening? Once the absorption process is occurring, so there is a starting of the metabolism. After being absorbed, these monosaccharides are getting absorbed in intestinal cell membrane, then the metabolism process will be initiated. After being transported from the intestinal tract, these monosaccharides are carried by a portal circulation directly to the liver. In the liver, most of the glucose is phosphorylated to glucose 6 phosphate. So here we can see this portal circulation of nutrient rich blood between the gut and liver is called portal circulation. And the remainder of glucose passes into the systemic blood supply. Any leftover any glucose is there, that is passing into the systemic blood supply. Other dietary monosaccharides or fructose, galactose are converted into the glucose in the liver. In the liver, glucose undergoes several metabolic pathways depending upon the supply and requirement of glucose to the body. And here also we can see this metabolic pathways of glucose in the liver, how it is getting this blood sugar glucose will be going for the glycogen and converting as a pentose phosphate with the help of pentose phosphate and NADPH with the help of acetyl coenzyme, and which giving the carbon dioxide and water. And we can see NADPH is indicating nicotinamide adenine dinucleotide phosphate, whereas acetyl coenzyme A. Here we can see the major metabolic pathways of carbohydrates are glycolysis, citric acid cycle, gluconeogenesis. Then two more is there, that is just like glycogenesis, glycogenolysis, and hexose monophosphate shunt. So let us summarize our topic. So digestion and absorption of carbohydrates, which is happening in the digestion in the mouth, stomach, in the intestine, and with the help of pancreatic enzymes, and digestion due to intestinal enzymes. So whereas absorption, mainly in the absorption process, we are notifying active transportation and facilitative transport. And finally, we have discussed regarding metabolism of carbohydrate in the major activities we can able to notify. I hope, my dear student, you understood the concept. Have a nice day. We'll meet once again.